investor call, and I'm happy to welcome you all and like to tell you a little bit about Nyrada. So, if you'll, Dimitri, if you go to the next slide. One of the nice things about Nyrada is it combines a board of directors and a scientific advisory board, which is used to playing on a very large global stage, and had a history of success in biopharmaceutical development. But we combine the great parts of the fertile soil of the Australian biotech industry, which includes a terrific regulatory regime, which allows early stage drugs to do phase 1 trials without FDA IND approvals. Second of all, a great tax regulatory regime, which uh, it allows very efficient use of capital to do those early stage animal studies and phase 1 trials. And uh, and and we also have U.S. capital markets expertise, with a number of us being from U.S. publicly traded companies. So we know how to make the migration when the time is right to be able to get the company to play on a global stage. So if we go on to the next slide, I'll talk a little bit more about the board of directors, which is, I'm a big believer that a great board of directors is a tribute to the power of the ideas of a small company like Nyrada, which has so much promise. So I'll start in the lower right corner, which is, I'll start with Chris Cox, who's one of our directors. He was the chief commercial officer of the medicines company, and which was sold for $9.7 billion to Novartis. And Chris is currently partnered with Ian Reed, who's the former CEO of Pfizer in a company called Population Health. And what really attracted Chris to join the board of Nyrada was, I've worked with them for the last 10 years, but he also saw a pattern where the success of the medicines company was they had partnered with the British National Health Service to do their clinical trials. And when he understood that we have a partnership with the U.S. Army Walter Reed Medical Center, which is the largest potential customer for our products, he said, "Hey, look, that seems like a winning combination to me." And he really believes in this company. Dr. Rudy Wieselow, who is employed at Merck KG, the German Merck, and he's done over 50 business development deals. So, eventually, when the time is right for us to license our products or partner, we have a world-class global expert in business development deals and terms. Marcus Frampton is the chief investment officer of the Alaska Permanent Fund, which manages $88 billion. And Marcus really has a reputation as a terrific biotech investor. His involvement is, he's invested with his own personal funds, but among the many companies that Marcus has helped create, one is Juno Therapeutics. It's named after a town in Alaska. They invested $250 million in it, and it was sold um, to Celgene and then eventually to Bristol Myers Squibb for $9 billion. So Marcus's involvement is terrific from a point of view of other institutional investors, and as we scale the company up, access to the capital markets. Dr. Gisela Mautner is a PhD MD. Um, she's the CEO of Noxopharm and very experienced. She previously was with Amgen and has worked on large-scale global projects like we intend to do as well. Dr. Ian Dixon is a successful Australian-based businessman, and myself, I've been in financing biotech startups for the last 20 years. So I've had some interesting successes and that contribute and can support James's great efforts. So with that, I'll hand the call over to James Bonner, our CEO.
Thanks, John. And I'd just like to extend a warm welcome to our shareholders and thank you for joining this call this morning, and in particular to new shareholders who just joined the journey fairly recently. So I hope you'll get something out of this call in terms of some more background on the company and where we're going. So by way of introduction, I've been working in biotech since the late 90s. So over 25 years now. My most recent role was on a drug development program at Neuron Pharmaceuticals. That was the development of the debut Rett syndrome drug, and it was during that program that it really sparked an interest for me in neurology research. And that is one of the key reasons behind me joining Nyrada when I heard about the work that was being done by Professor Gary Housley and his team at UNSW, and the potential to work on another brain injury program. So just before I sort of start, I just wanted to make a few general comments about the biotech sector in general. The last couple of years have been fairly tough for biotech. And those who follow the sector will know that sentiment's been fairly poor. There have been lots of layoffs, aborted programs. Deal making has activities sort of dried up. But we think that there are, despite not being economists, that there are signs of a turnaround, and we're starting to see some activity and improvement in sentiment. I think one thing I would say is that the companies that have got through the challenges of the last couple of years and come out of it well are companies who have focused on running efficient operations and using smart allocation of capital. So that brings me to Nyrada and where we are, and I think we're in a fairly fortunate position. First off, I think we're operating in a region that has been shielded to a large degree from some of the headwinds that other companies have faced around the globe. Australia is a very good place to do early stage research and clinical research, and I think if you look at the things that we can control, we've done a fairly good job of that. We've focused on what we believe are two high value programs. And we've tried to build into our thinking and our strategy optionality and contingency, and balancing the risk of having a backup program should something go wrong, and balancing that against spreading ourselves too thinly. So we're going to talk mainly about the brain injury program today. And that follows on from results we announced at the end of February, very positive results. So we'll talk about that. I think the other thing I'd say about Nyrada, and it's helped us weather the storm, is that we're a very efficient, lean operation. We have very low overhead costs. We maintain a small team of 5.5 FTEs. And we operate like a virtual company, so the operation is adaptable and nimble and easily scalable. I think we'll move on from there, Dimitri, and maybe talk about the financials.
Sure. So the way we'll run this presentation today is, rather than James go through a slide deck, we'll pose a couple of questions to James, and we'll give him free range to speak, and then investors can ask anything at the end should there be a gap. So the first question to James is, Nyrada has just released its 3 quarterly numbers. What are the highlights?
Sure, let me just bring up the slide for that. So following on from what I've just been talking about, I think Nyrada is in a good position in terms of its cash situation. In the latest quarterly, we announced 5.6 million in cash, and that follows a successful fully subscribed placement that was conducted during March. We raised $1.7 million, and there's a further 200,000 to come in following a shareholder approval at the EGM that Dimitri mentioned. We've also received a fairly substantial cash injection in the form of an R&D tax rebate that came in at the end of last year. We're expecting another one to come in at the end of this year. It's difficult to predict the quantum of that rebate, but it's based on 42.5% of R&D spend. And in the last year, we've spent about $6.1 million. So as I said, it'll depend on how much we spend on R&D in this current financial year. So overall, I think the cash position is healthy, and we're in a good position to take the brain injury drug forward into a phase 1 study, which we're going to be running in the second half of the year. Dimitri?
Do you want to change the slide? Okay, the next question. Next question is, what are Nyrada's key development programs?
Sure. Well, I mentioned we had two high-value programs. I'll just talk briefly about the cholesterol-lowering drug program. We announced in the second half of last year, calendar year, unfortunately disappointing results from a toxicology study with our PCSK9 inhibitor. We were hugely disappointed with that result because we really felt that drug had huge potential. We still believe that a small molecule PCSK9 inhibitor is the optimal treatment for lowering cholesterol. And we're still working on that program, doing low-cost background work, fairly much sort of a paper research exercise. But the key focus of the company currently is the brain injury drug, NYRB-003. And that was the drug that we recently released results from. Now both the programs we're pursuing, it's based around Australian-developed innovation and technology. The brain injury drug was developed in conjunction with the University of New South Wales here in Sydney, and Professor Gary Housley, who is the principal scientist and chair of our scientific advisory board. Now, what's exciting about NYRB-003, and I'll come to the data shortly, is that this is a novel molecule. It's never been made or tested before, and we believe it's got the potential to be a first-in-class treatment for brain injury. And when I talk about brain injury, I'd just like to say that the pathology in brain injury in terms of stroke and traumatic brain injury, if you think of blows to the head and falls, motor vehicle accidents, etc., is common. So one of the exciting aspects of this molecule is that it has the potential not only to be used in stroke, which we've now got good efficacy data for, but also to be used in traumatic brain injury and potentially other neurological indications as well. When I say first in class, currently there are no FDA approved drugs to treat secondary brain injury, the indication we're going after. Yeah, it's an exciting prospect because we believe we're the only company in the world that's developing a drug that acts on this particular target, which I'll talk about shortly.
Okay, James. Nyrada recently released the results of its stroke study, a brain injury program stroke study. Can you tell us about that study, please?
Sure. So the study that we've just completed and reported on, it was in a model of stroke, in a mouse model of stroke. And just as an aside, this model was actually developed by Professor Junichi Nabakura, who is also on our scientific advisory board. This mouse model of stroke is considered one of the two sort of gold standard models that are used to test potential stroke drugs. And in this study that we've just completed, we were able to show that we achieved a 42% neuroprotection with the drug versus animals that received vehicle or placebo, if you like. So that was a very strong signal. It exceeded our expectations. One of the key features of this study was that we used a technique for imaging the brains in these mice using a small animal MRI instrument that UNSW have. It's specifically designed for imaging small animal brains. And it was using a technique that we developed in conjunction with the Walter Reed Army Institute of Research in the US. This is the DoD department that looks specifically at traumatic brain injury research. So we're able to use the findings from that pilot study and apply that technique to imaging the brains of these mice in this study. So it's fairly cutting edge stuff. In addition to showing that the drug is efficacious in reducing injury size, we also looked at a biomarker, which is a marker of injury to axons in the brain. Now, axons, if you think about the neuron being the computer, the axon is like the data cable that transmits the data around the body. And NFL is a biomarker for injury to those axons. And we got good corroborating data in this biomarker where we achieved a 41% reduction in this injury biomarker level. So these mice in the study, they were treated for 72 hours via intravenous infusion. And that's the way we intend to administer the drug in the clinic. And I'll come back to that in a little bit. But one of the other outcomes of the study was that in addition to showing strong efficacy, we also showed or demonstrated good tolerability. So that bodes well for the safety studies that we're currently undertaking. Okay.
Yep.
So I mentioned briefly secondary brain injury, and this is what the drug is being developed to address. And if you look at this slide here, you can see on the right-hand panel a representation. In fact, this is an actual scan of a rat brain showing what secondary brain injury looks like. So if you look at the top image there, you'll see after 30 minutes you get this what's called the primary injury. This is a penetrating injury, and this work was done at Walter Reed, and this mimics what would occur from, say, a bullet wound or a shrapnel wound. You can see the brown area that represents the primary injury, and if you look at the image below that, you'll see that after 72 hours, that injury is expanded out significantly, and that's the phenomenon called secondary brain injury, and that's in essence what this drug is developed to address or reduce. And if we plot that on a graph, you can see there on the left-hand side, you've got the primary injury in blue, you've got the secondary injury in green, and if you reduce that secondary injury, the aim is to reduce the risk of mortality and also limit the level of disability and improve recovery time, etc. Now, I'm going to spend probably less than a minute on some cell biology here, because I know that puts a lot of people to sleep. But this essentially is how the drug works. The drug blocks a family of ion channels called TRPC channels. And what you can see presented here, if you look at the gray dots, that represents the surface of a neuron or a brain cell. And what happens following a brain injury, it could be a stroke or it could be a traumatic brain injury, is that you get a massive release of a neurotransmitter called glutamate. And that glutamate, which is shown here as the pink trapezoid, it binds to its receptor, and that causes a chain of events that leads to a condition called excitotoxicity. Now, these ion channels, these TRPC ion channels that our drug has been developed to block, they open and let an ion called calcium into the cell. And that is fine under normal circumstances, but following a brain injury, where you've got such a high release of glutamate, it causes excessive calcium to enter the cell. These channels, which are shown here as the vertical bars, they basically open up and let all that calcium in. And that calcium builds up to toxic levels and it kills the brain cell. So that's essentially what we're trying to address here. We're trying to keep those ion channels in their closed position to block that calcium entry. So that calcium dysregulation leads to neuronal cell death, and there's also evidence to suggest, and this is yet to be shown, that it can also lead to neuroinflammation through its action on other cell types in the brain as well. So that's the end of the cell biology. So why do we think this is going to work? And well, we've shown it works, but the genesis of the program really started out in the late 2010s with a key paper that was published. And in this study that's presented here, there was a study done in a what's called a genetic knockout mouse. So these are mice that don't have these TRPC channels that you saw on the last slide. And this study was again a stroke study. It was slightly different, but again it was in mice. And you can see if you look at the brain slices in the left panels there, that when those channels are knocked out and the animals are given a stroke, they're protected. So the WT stands for wild type. They're normal standard mice, and you can see that the area of injury, which is shown as the white area, that's dead brain tissue, is much larger. And that's been quantified in the graph there in the middle. And so with these knockout mice, you actually get neuroprotection, and you get about a 50% improvement, as you can see by this bar graph, over wild type mice. So the scale on the left there is injury volume. There's also some additional data that we haven't presented here, which shows that these mice also performed better when they underwent neurological assessment. So there's a score called a neurological disability score, and the TRPC knockout mice perform much better. So this was really what got us started on this program, and the objective all along has been to mimic what happens in a genetically knockout mouse with a drug. And I'm just flashing up here the results of the study again. So the 42% reduction is fairly close to what can be achieved when you completely knock those channels out. So that's what's got us really excited. Plus also the corroborating data with the injury biomarker. Now there is some further work that we're doing as part of the same study. We are currently imaging the brains of these same mice for other injury biomarkers. It's a process called immunohistochemistry. It's exploratory, and we're not sure what we're going to find, but we're currently doing that work at UNSW, and that'll provide us with some more information about the mechanism and corroborate the data we've got here. So we'll be announcing the findings of that work once it's completed.
Okay, James. What is the market opportunity for NYRVI-03?
So I mentioned that we're going after two indications. We've got efficacy data in a clinically relevant stroke model. We've also got some work that we're doing with the Walter Reed Army Institute of Research, where they'll be looking for an efficacy signal with the drug in a penetrating brain injury model, traumatic brain injury model. So one of the attractions with this program, or what's getting us excited, is that it has the potential to move into two indications. Now, the first of those is stroke, and stroke presents a huge market opportunity. About 15 million people suffer a stroke annually each year. In Australia, about 100 strokes occur each day. And the treatment options for patients who sustain a stroke are fairly limited. One of the frontline treatments is a class of drug called the TPA or tissue plasminogen activated drugs. And these effectively dissolve the clot following an ischemic brain injury or stroke. Unfortunately, however, there's a very narrow time window that these drugs have to be administered within, and it's about 4.5 hours. And so consequently, only about 15% of patients receive this treatment. So the other 85% miss out, unfortunately. Just to sort of put into perspective the market for the current drugs that are available for stroke, the TPA drugs, the per treatment cost is between $7,000 and $10,000 per treatment, and the TPA drug market last year was worth about $2.5 billion. So given the relatively small number of patients treated, it's a sizable market. Now, just one thing about the drug we're developing, we don't anticipate that there's going to be this golden treatment window. We believe that as long as the drug is administered within the time that secondary brain injury occurs, which is around 72 hours, patients will see some benefit. The problem with the TPA drugs is that if they're given outside that 4.5-hour window, they can cause what's called reperfusion injury, which can be dangerous in itself. So plenty of potential in the stroke market. The traumatic brain injury market is an interesting one because there are currently no drugs approved to treat traumatic brain injury. So when you look at the current stats on sales, they're really for drugs that are supportive. If you think of anticonvulsants and sedatives and so on, but there is no drug currently available to prevent secondary brain injury. But we know the numbers are very large. And one thing we are doing is to pursue what's termed as moderate to severe traumatic brain injury initially. And there are a number of reasons for doing that. The first is that the dose form that we've developed is an intravenous dose form, and it can be given via infusion over 3 days. We are planning to develop an oral or intranasal dose form for milder forms of TBI, like concussion. But for patients who suffer a severe traumatic brain injury, they're usually inpatient, and an IV dose form is considered advantageous or preferred. The other thing about moderate to severe TBI as well is it comes under the classification of orphan drug. So there are benefits for developing an orphan drug, like an accelerated regulatory pathway. You get tax breaks on clinical trial costs and so on. So that's the approach we're taking. And I would say that the treatment approach, the 3-day infusion, would be the same for patients who sustain a traumatic brain injury and for patients who sustain a stroke. So just finally on that, I think the markets for moderate to severe TBI and concussion are quite different. I think the amount of money that you could reasonably charge for a drug to treat a serious and life-threatening condition is in a different ballpark to, say, a drug to treat a mild TBI or concussion. So we're going to make sure that we don't cannibalize, one market cannibalizes the other.
Before we get to the next question, James, I'd encourage participants to post any questions they have. We've got a couple of questions. We'll get to them shortly, but if you have any questions, please put them in the chat and we'll get to them shortly. Last one from this end is, what are the key coming milestones and the future for BIO3, James?
Sure. So following the successful completion of the efficacy study, we're now doing what's called GLP, or Good Laboratory Practice studies. Now, these are regulatory mandated studies that have to be completed before first-in-human trials can be initiated. And that work is currently underway. It'll take 3 to 4 months, and we're planning to have a quick transition from those studies into phase 1. So the phase 1 study is planned to start second half of the year. The plan beyond phase 1 is to open up INDs, or investigational new drug applications, with the US FDA, and then pursue phase 2 development in the US. Now, the reason we're doing it in the US, there are a number of reasons, in fact. It gives you access to a larger patient population, so that is important in terms of recruitment rates, although it would be great to have a couple of sites here in Australia as well. The other thing too, and this is in particular related to traumatic brain injury, is that the US, there's the potential for attracting non-dilutive funding for clinical research from phase 1 and beyond. And we feel that we've got a better chance of taking advantage of that if we run those studies in the US. But I think the main driver is really just the timelines and getting phase 2 studies completed as quickly as possible. The phase 1 study, which I'll talk about now. Just bear with me a second. The phase 1 study will be run here in Australia. And this will be a healthy volunteer study. I think what's interesting about the development strategy for this drug is that this phase 1 study will support going into a phase 2 study for either stroke or traumatic brain injury. So we get a lot of bang for our buck out of this phase 1 design. It'll be in healthy male and female volunteers, and the design of the study is fairly standard. It's called a single ascending dose, multiple ascending dose study, where we'll start off giving single doses of the drug in an escalatory fashion, and following that, we'll then give the drug for 72 hours as a continuous infusion, which is the way we intend to dose in the clinical studies, phase 2 clinical studies. So, like I said, that study will get underway in the second half of the year, and once that's completed, we'll open INDs with the FDA and have a quick transition into phase 2.
Okay,
and that includes everything I wanted to say, Dimitri.
Again, we have a couple of questions which I'll read out in a second. So if you have anything to ask, please do. Just one quick one, James. Is the phase 1 dependent upon the Walter Reed study?
So the Walter Reed study, unfortunately, there's been a slight delay in getting that underway due to key staff unavailability. But that's planned to get underway shortly, and it's not on the critical path for phase 1. It'll run concurrent with the GLP studies. And the phase 1 study is not dependent on that.
Okay. Questions from Paul Hart. When do you expect the TBI program with Walter Reed to commence? You've already addressed that. And are they funding the work? Walter Reed, that is.
Yeah, so there is some funding that we're contributing. It's really to cover the cost of the study. Yeah, but the work's being done under an agreed CRADA, which is a collaborative research and development agreement. So the cost we're paying is a small cost, and it's really just to support the work.
Next question. Are the hurdles for FDA approval lower, given there is no treatment for stroke or TBI?
That's a great question, and that's really one of the drivers for going after orphan drug designation and looking at moderate to severe TBI first. I think, from my experience of running a traumatic brain injury study, this is back in my Neuron days, under FDA control. There's a good opportunity with a severe and life-threatening indication for a favorable regulatory pathway, let's say. So there are some programs within the FDA, like accelerated approval, fast track approval, the possibility of getting away with a single pivotal phase 3 study. So it's very much an accelerated pathway for these sorts of drugs.
Next question. When do you expect the additional work on biomarkers currently underway to be completed?
So the imaging has been completed, and it's at the data analysis phase at the moment, but it is fairly tortuous and arduous and long-winded, because there are literally hundreds of brain slices that need to be individually quantified and data collated. So we're pushing that forward as quickly as possible, but it's hard to put a firm date on it at this time. But it's in the near term.
Next question, and I think it builds upon the earlier one. Will Walter Reed provide financial support for TBI? And I think the reference possibly means to non-dilutive or other funding sources.
So Walter Reed have already contributed a lot in terms of in-kind support. We did an extensive pilot program, and that was really a good springboard for doing the recent stroke study, where we applied the knowledge from that pilot study. But in terms of non-dilutive funding going forward, there is non-dilutive funding available through the US Department of Defense, but not Walter Reed specifically. And just on non-dilutive funding, there's a group that was formed. It's a consortium between the FDA, industry, and academia. They were set up a few years ago to look at how traumatic brain injuries were being studied, and a lot of the funding from the US Department of Defense went to support the work that they did. So they were looking at things like, how MRI is conducted, how clinical trials are designed. They did a lot of work on blood biomarkers of injury. I mean, you can Google Track TBI Network and find out more about the work that they're doing. But it's really foundational work to support the development of a drug like NYRBIO3. So I think if we're successful and can get through phase 1, I think there's a good chance that we'll get support in some form to take that forward, because the DoD are very clear that traumatic brain injury is a key health concern for them, and they're very keen to come up with a treatment.
The next question, and I might interject on, answer on your behalf. They ask, what is the estimated cost for the GLP studies and subsequent experiments? And I think the answer on that one, we don't disclose to that level of granularity. There's a level of confidentiality to that because we're working with partners. However, you can find the macro information in our regular financial disclosures. Unless you want to add anything to that.
Yeah, no, that's fine, Dimitri. I mean, those costs can be Googled, I guess, but I don't have those numbers available to me anyway, so I'd be probably putting you wrong. But
the last question for the moment, unless there's any furthers, is what is the difference between Nyrada and Argenica's drug and trial? Other competitors, thanks.
Well, there aren't too many competitors. There are a couple of drug programs that I'm aware of that are pursuing a peptide approach to treating stroke and traumatic brain injury, and I believe the Argenica molecule falls into that category. It's not really up to me to comment on other companies' programs. I mean, I'm very much focused on our approach. Our approach is slightly different. We're developing a small molecule, and I think one of the key things that I should also say about our drug, it's been developed specifically to cross the blood-brain barrier. So, one thing that is a big challenge in neurology drug research is getting the drug to the target. And this molecule that we're developing, NYRBIO3, has been developed from the ground up with a specific target and role in mind. We've shown through numerous studies that it really crosses the blood-brain barrier. So, but how it compares to other drugs that are being developed, like I said, there aren't that many in development. So we're really at the cutting edge of this, yeah, it's not up to me to provide an assessment on other companies' programs.
Are there any further questions? Because if not, we might wrap it up. I'll just wait a little bit to see if there are any further questions. Well, that appears everything. So James and/or John, if you have any closing comments, or not. Otherwise, we'll wrap this up. Thank you for everyone's attendance.
No further comments from me. Just one thing. There's a video animation on the last slide, and if I put you to sleep during the cell biology part of the presentation, you can go back and have a look at that. It's a very simple way of explaining how the drug works. But thanks everyone for joining. Appreciate your interest.
And by the way, Dimitri, Dev Ramachandra asked if this recording of this presentation is going to be on our website.
I will look into that. We don't have it.
Great.
I will endeavor to do that.
Great. Well, thank you, everyone. We get by with little help from our friends.
Thank you all
Thanks for listening.
your attendance.
Thanks all.
Appreciate your support. Goodbye.